It's like in those crime shows, right? You catch the suspect, but the case isn't really closed. You still got to worry. What if there's like a whole network out there ready to make their move? That's kind of what we're dealing with today. Circulating tumor cells. CPCs. The silent spreaders in the big mystery of cancer.
Wow, that's a really good way to put it, because isolating these CTCs, it's a huge challenge. Think about it. They're outnumbered by normal cells by like billions to one.
Billions to one. So trying to find these things.
It's like searching for a specific grain of sand on an entire beach.
Okay, yeah, I get the picture. So in our source material, this paper we've got, it's all about this technology called eDAR. And they're making it even better at finding these, what did you call them? CTCs. Can you break down this eDAR thing for us? What is it exactly?
Sure. So eDAR stands for Ensemble Decision Aliquot Ranking. Basically, it's this super high-tech sorting system. It uses lasers, microfluidics, all that good stuff to capture these rare cells.
Okay, so we're talking like way more precise than just a regular filter.
Oh yeah, way more advanced. Imagine a filter that can tell the difference, not just by size, but by these tiny little markers on the cells themselves.
Okay, so it's looking for something very specific.
Yeah.
The paper mentions the purity of the samples they get with eDAR a lot. Why is that so important?
Well, imagine you're trying to, let's say, study a single grain of sand,
Right.
but it's mixed in with a whole bucket load of other sand.
Mm-hmm.
It'd be really hard to figure out anything about that one grain. It's the same with these CTCs. If you've got too many other cells mixed in, it's like...
That's noise.
Exactly. Too much noise.
Yeah.
You need that pure sample to get a clear signal, and that's crucial for accurate analysis.
Makes sense. And it seems like these researchers, they're really focused on that. They added a whole second sorting step to their eDAR system. What's the deal with that? Is that like a quality control thing?
You got it. Think of it like panning for gold, right? The first sort, that's like the initial sift, gets rid of the big chunks of stuff you don't want. But then that second sort, that's where you're really carefully looking for those golden nuggets, making sure you're only capturing the most valuable stuff. In this case, that's the CTCs.
Oh, I like that analogy. So double-checking their work, basically. Okay, so they're getting really good at grabbing these CTCs, but then they talk about aliquot stretching, and that honestly sounds like something you'd do in a yoga class, not a lab. What is that?
Yeah, right. It's a funny name, but it's all about how they're manipulating the fluids inside the eDAR system. They're using these things called herringbone features inside those tiny channels. You could think of them like...
grooves, almost.
Yeah, like grooves strategically placed along the channels. And these grooves, they're not just randomly placed. They actually create these tiny little whirlpools in the fluid as it flows through.
Wait, whirlpools inside this tiny little device? How does that even work?
It's pretty wild, right? But those little whirlpools, they help to space out the cells even more as they're flowing through. Kind of like creating a little more breathing room in that channel, and that makes it even easier to separate out those target cells.
So those grooves are like what? Directing traffic inside the eDAR system? Making sure the CTCs don't have any friends too close by?
Exactly. It's all about creating the perfect environment to isolate those cells.
Wow, okay, so we've got double sorting, we've got strategic grooves, we've got tiny little whirlpools, all working together to catch these CTCs. What kind of results are we talking about here? How much better does this actually make things?
Well, get this. They were able to get a 70-fold increase in the purity of their isolated CTC samples compared to the older methods. That's huge.
70 times better. That sounds like a game changer. What kind of possibilities does that open up? I mean, that's got to be huge for research.
Oh, it's huge. I mean, with samples this pure, now we're talking about being able to really dig into those CTCs, analyze their DNA, see what makes them tick.
So this is how we start to figure out how cancer spreads and, maybe even how to stop it.
Exactly. And not only that, think about early detection. Right now, we're usually playing catch-up with cancer.
Yeah.
But if we can find these CTCs in the bloodstream before a tumor even shows up on a scan...
Oh, that would be incredible. We're talking about catching cancer before it even really becomes cancer.
That's the dream.
Yeah.
It would completely change the game.
No kidding. Okay, so earlier you mentioned something about these CTC clusters. I wanted to circle back to that. What's the deal with those?
Yes. The clusters. This is where things get really interesting. See, we've been talking about these CTCs like they're, lone wolves. But what if they're not? What if they're actually working together?
Okay, so it's not just one bad cell escaping. You're saying there's a whole gang of them causing trouble.
Something like that. There's a growing body of research that suggests that these CTCs, they might actually be communicating with each other, coordinating their movements. Maybe even,
Designing their attack. This is wild.
right, it's like they're forming these little raiding parties, spreading out to establish those new tumor cells.
Okay, so we've gone from stealthy spies to a full-on invasion force here. And you were about to say something about a leader.
Oh yeah, some researchers believe that within these clusters, there might actually be a leader cell calling the shots, so to speak.
A leader cell, directing the others. Okay, now this is straight out of a sci-fi movie. We're talking about intercepting messages between enemy cells now. This is amazing.
It's definitely a new frontier in cancer research. But that's what makes this research so exciting. If we can figure out how these clusters work, how they communicate, how they choose their targets,
We could figure out how to shut them down.
yeah, potentially disrupt the whole system.
Okay, so instead of just looking for these individual CTCs, now we're trying to, like, crack the code of their secret communication network.
Exactly. And it's not going to be easy, let me tell you. These CTC clusters, they're even rarer than those individual CTCs we were talking about.
Oh, I bet. Way harder to find. It's like, It's like trying to find, I don't know, a rare flower. That only blooms for like an hour in the middle of the night,
but
at the
like,
top of a mountain, somewhere. You get the idea.
yeah, pretty much. But hey, that's what makes this eDAR system so exciting. With this, we're actually getting closer to being able to capture these clusters, study how they behave.
This feels like, I don't know, a real turning point. Like we
Yeah.
finally have the right tools to unlock this whole stage of metastasis, you know?
Yeah, I think that's a great way to put it. And if we can actually pull this off, I mean, the potential payoffs are enormous. Imagine being able to, like, interfere with the communication within these clusters.
So they can't coordinate anymore.
Exactly. Cut off the mastermind from the rest of its network.
And that could stop metastasis before it even starts.
It's possible. Still a lot of research to do, but yeah, the possibilities are incredibly exciting.
Well, this whole deep dive has been amazing. We started with these sneaky little CTCs, and now we're talking about, like, intercepting messages in their little evil hideouts.
It just goes to show, cancer is incredibly complex, but the researchers who are studying it, they're even more incredible, always coming up with new ways to understand it, to fight it.
Totally. So to all our listeners out there, if you're feeling a little overwhelmed by all this, don't worry, it's a lot. But hopefully you're also feeling, a little bit hopeful, because there's some amazing research happening right now.
Every new discovery, every new technology, it's all bringing us closer to figuring out this disease, finding better ways to treat it.
Couldn't have said it better myself. And this research on eDAR, on these CTCs, that's a perfect example. They're pushing the limits of what we thought was even possible, opening up whole new paths for exploration.
And they say sometimes the most groundbreaking discoveries, they happen when you're exploring something totally new.
So true. And on that note, we're going to wrap up this deep dive, but keep those questions coming, folks. Keep that curiosity alive, because who knows, maybe you'll be the one to make the next big breakthrough.